There is one molecule in your body that does something no supplement, no medication, and no amount of willpower can do. It physically triggers the growth of new connections between your brain cells. It is called BDNF, brain-derived neurotrophic factor. John Rady at Harvard Medical School, who has spent his career studying its effects, calls it miracle grow for the brain. Here is what makes this video different from the last two in this series. The first explained what happens to your brain after 40. The second explained why neuroplasticity does not actually decline, only the conditions for it do. This video gives you the single most powerful input for creating those conditions. And it is not what most people think it is. There is a specific protocol, tested in a landmark study that ran for an entire year, that produced a measurable increase in the size of the hippocampus in older adults. Not stabilization, growth. And the mechanism behind it starts somewhere you would not expect. Your muscles. BDNF is a neurotrophin, a type of protein that supports the survival, growth, and connection of neurons. It is produced primarily in the hippocampus, your primary memory structure, and in the cortex, where higher-order thinking happens. Here is its specific job. When BDNF is released, it binds to a receptor called TARKB on the surface of neurons. This binding activates a cascade of intracellular signals that does three things. It promotes the survival of existing neurons. It supports synaptogenesis, the formation of new connections between neurons, and it enhances long-term potentiation, the exact mechanism from the previous video in this series. In other words, BDNF is one of the direct molecular drivers of neuroplasticity. Higher BDNF levels are associated with larger hippocampal volume in multiple studies. Lower BDNF levels are associated with increased risk of cognitive decline. And here is the part that connects directly to everything in this series. BDNF production declines with age. It also declines with chronically elevated cortisol, the exact mechanism described in the previous video. The two problems compound each other. Lower BDNF means less capacity to build new connections. Higher cortisol means the connections that could be built are actively suppressed. Here is the study that changed how researchers think about exercise and the aging brain. Kirk Erickson and colleagues recruited healthy older adults and put them through 1 full year of moderate intensity walking. 3 sessions per week, 40 minutes per session, heart rate at 50 to 75% of their reserve. Not intense, moderate. After 1 year, the walking group showed a significant increase in hippocampal volume. Not a slowing of decline, an increase. And the increase was closely associated with elevated serum BDNF levels. This was not a young population. These were older adults. The exact population everyone assumes is past the point of structural brain growth. Their hippocampus grew anyway, because the input was right. Here is the mechanism that explains why exercise specifically, more than any other intervention, drives BDNF production so powerfully. In 2013, a research team led by Christian Rahn published findings in the journal Cell Metabolism that identified the specific signaling pathway. When your muscles contract repeatedly during sustained exercise, they activate a protein called PGC1 alpha inside the muscle cells. PGC1 alpha then increases the production of another protein called FNDC5. FNDC5 gets cleaved and released into your bloodstream as a hormone called irisin. Irisin is what researchers call a myokine, a hormone released by working muscle that travels through the blood and acts on other tissues. One of irisin's primary targets is the brain. When irisin crosses into your brain tissue, it triggers an increase in BDNF expression, specifically in the hippocampus. Your muscles, when they work hard enough for long enough, send a direct biochemical signal to your brain telling it to grow. That is not a metaphor. That is the literal mechanism. Movement is not just good for your body. It is a direct line of communication to your brain's growth machinery. This explains why exercise outperforms almost every other intervention tested for cognitive health in aging populations. It is because muscle contraction specifically triggers a hormonal cascade that ends in BDNF release in the exact brain region most vulnerable to age-related decline. Think about the last time you went for a genuinely brisk walk and then sat down to think or work afterward. Remember how your thinking felt? Sharper, clearer, like the static had cleared. Most people have felt this and called it a mood boost. It is not a mood boost, it is BDNF, at its peak concentration in your bloodstream and your brain tissue, doing exactly what it is designed to do. You have already experienced the mechanism this video is describing. You just did not have a name for it. Quick honest check before the protocol. How does your current exercise pattern compare to the Erickson study protocol? 1. I do not currently have a consistent aerobic exercise routine. 2. I exercise but inconsistently, maybe once a week or less. 3. I exercise 2 to 3 times a week, but rarely at a sustained moderate intensity. 4. I am already doing something close to 3 sessions of 40 minutes at moderate intensity weekly. Drop your number in the comments. Before the protocol, I want to address 2 common mistakes. The first is intensity confusion. A 2020 study by Renteria and colleagues found that short-term high-intensity interval training increases systemic BDNF specifically in healthy women. So HIIT works too. But the Erickson study, the one that produced measurable hippocampal growth over a full year, used moderate intensity. Moderate, sustained aerobic activity should be your foundation. The second mistake is thinking this is exclusively about cardio. Resistance exercise also triggers growth factor release, including BDNF and IGF-1, supporting structural and functional plasticity in the hippocampus and prefrontal cortex. The strongest evidence base is for aerobic exercise, but resistance training adds a complementary pathway, not a competing one. Now stop. Before the full protocol, one finding that changes how you should structure your day. The BDNF increase from a single exercise session is not permanent. It is acute. It rises sharply during and immediately after exercise and begins returning to baseline within roughly 30 to 60 minutes. This means there is a specific window, immediately following exercise, where your brain's capacity for forming new connections is at its daily peak. If you have something important to learn, a complex problem to think through, a skill you are trying to build, the 30 minutes after you finish exercising is the single highest leverage cognitive window in your entire day. Most people exercise and then immediately move to low-demand activity. They are wasting the single highest plasticity window biology gives them each day. Here is the complete BDNF protocol, built directly from the strongest available research. Foundation: moderate aerobic exercise, 3 times per week, 40 minutes per session. This is the exact structure from the Erickson study that produced measurable hippocampal volume increases over 1 year. Walking at a pace where you could hold a conversation but would not want to sing. Heart rate at roughly 50 to 75% of your estimated maximum. Addition: 1 to 2 sessions of resistance training per week. 2 sessions of 20 to 30 minutes targeting major muscle groups is sufficient to engage the complementary growth factor pathway. The critical addition: use the 30-minute post-exercise window deliberately. Immediately after your aerobic session, while BDNF is at its peak, spend the available window on your highest value cognitive task. Learning something new. Working through a complex problem. This is the window where your brain is most receptive to forming new connections. Together, this protocol addresses both conditions from the previous video in this series. The exercise lowers cortisol, supporting the cortisol window for plasticity, and it directly produces the BDNF that drives the synaptogenesis and LTP enhancement that makes new learning physically possible. Here is what the research timeline actually looks like if you start this protocol today. Within the first session, an acute BDNF spike, peaking shortly after exercise. Within the first 2 weeks, many people report the subjective clarity described earlier becoming more noticeable and more consistent. Within 3 months, measurable improvements in executive function have been documented in exercise studies of this duration. Within 1 year, the Erickson study's actual finding: a measurable increase in hippocampal volume. This is not a quick fix. It is also not a multi-decade uncertain hope. It is a documented, replicated 1-year timeline to measurable structural brain growth. Available to you, starting with the next walk you take. Exercise is not for your body. It is for your brain. Your body is just the mechanism your brain uses to ask for what it needs. Every contraction of working muscle is a direct biochemical message telling your brain to grow. That message does not stop being sent at 40. It is still being sent right now. The only question is whether you are sending it. This is the third video in this series, and it closes the arc the first two opened. The first video explained what changes in your brain after 40. The second explained that neuroplasticity itself does not decline, only the conditions for it do. This video gives you the single most powerful lever for creating those conditions, BDNF, and the specific muscle-to-brain pathway that produces it. 3 sessions of moderate aerobic exercise per week, 1 to 2 sessions of resistance training, and the deliberate use of the 30-minute window after exercise for your highest value cognitive work. Save this video. The next time you consider skipping a walk because you are tired or busy, remember what that walk is actually doing. It is not burning calories. It is sending a direct signal to your hippocampus to grow. Share this with someone who exercises purely for their body and has never been told what it is doing for their brain. That reframe changes the entire relationship with movement. If you are not here to stay average, subscribe.